Cardiovascular physiology, microcirculation, a deep dive into the exchange system of the human body. Welcome to our podcast on cardiovascular physiology, where today we take an in-depth look at microcirculation, the ultimate destination within the cardiovascular system. This is where the primary function of circulating blood truly unfolds, the vital exchange of materials between the blood and our body tissues. The core principle, the exchange vessels. Microcirculation refers to the intricate network of small vessels within our tissues, responsible for the exchange of gases, nutrients, waste products, and hormones. Think of it as the functional unit at the heart of the circulatory system, where the real business happens. Components of the microcirculation. Let us break down the microcirculatory bed, which consists of a carefully arranged series of vessels, each designed for a specific function. First, we have the arterioles. These are the smallest arteries, composed of 1 to 5 layers of smooth muscle. They serve as the primary source of resistance within the circulatory system, making them the main regulators of total peripheral resistance and, as a result, systemic arterial pressure. By narrowing or widening, a process known as constriction or dilation, they control how much blood enters each capillary network. This makes them the gatekeepers of blood flow, ensuring that blood enters the delicate capillaries at a lower, safer pressure. Next are the medarterioles. These vessels taper down and are partially muscular, containing scattered smooth muscle cells. Their main role is to provide a direct but regulated pathway between arterioles and venules. The capillaries follow. These are not only the smallest but also the most numerous vessels in our bodies. Their walls consist of a single layer of endothelial cells resting on a basement membrane, which keeps the distance for diffusion as short as possible. Capillaries are the primary site for the exchange of substances. This is truly the business end of the circulatory journey. After capillaries, blood enters the venules, the smallest veins. Initially, these vessels have little to no smooth muscle, but as they enlarge, they gain more. Venules are slightly involved in resistance and, in fact, can be even more permeable than capillaries, especially to larger molecules. This quality is crucial during inflammation and when immune cells need to migrate out of the bloodstream. Venules also act as a reservoir, holding a significant volume of blood due to their high compliance. Regulation of flow through the capillary bed. Blood flow within each capillary bed is not constant but is dynamically adjusted based on the needs of the tissue. One fascinating phenomenon involved is vasomotion, the rhythmic contraction and relaxation of medarterioles and precapillary sphincters. This causes blood flow in the capillaries to be pulsatile rather than continuous. Precapillary sphincters are rings of smooth muscle found at the entrance to each true capillary, right where it branches from a medarteriole. Acting like stopcocks, these sphincters control whether blood flows into a particular capillary. Their activity is mainly determined by local metabolic factors. The regulation of blood flow relies on several mechanisms. The most significant, especially for active tissues, is the metabolic theory. When a tissue becomes more active, such as muscle during exercise, its oxygen levels decrease, and it begins to produce metabolic byproducts like carbon dioxide, hydrogen ions, potassium ions, lactate, and adenosine. These factors cause arterioles to dilate and precapillary sphincters to relax, boosting capillary flow to meet the tissue's demands. A beautiful example of the body's autoregulation. There is also the myogenic theory, which explains how blood vessels react to changes in pressure. When the pressure inside an arteriole suddenly increases, the vessel wall stretches, and the smooth muscle contracts in response. This keeps capillary flow and pressure relatively constant, even when systemic pressure fluctuates. Endothelial cells lining the blood vessels also play a crucial role by releasing various substances. These include vasodilators such as nitric oxide and prostacyclin, which help widen the vessels, and vasoconstrictors like endothelin-1, which cause them to narrow. Finally, hormonal and neural controls are at play. Sympathetic nerves usually cause most arterioles, except those supplying the heart and brain, to constrict, helping to maintain central blood pressure. Hormones like epinephrine and angiotensin II also promote constriction, further fine-tuning blood distribution. Mechanisms of capillary exchange. Now let us focus on how substances actually move across the capillary wall. There are 3 primary mechanisms at work. First, diffusion is the dominant method. Here, molecules move from areas of higher concentration to areas of lower concentration. Lipid-soluble substances, such as oxygen, carbon dioxide, and steroids, pass directly through the cell membranes of endothelial cells very rapidly. In contrast, water-soluble substances such as ions, glucose, and amino acids move through water-filled pores or clefts between the cells. The size and number of these pores vary by tissue. For example, they are very tight in the brain, forming the blood-brain barrier, and much looser in the liver and kidneys. Next is bulk flow, also known as ultrafiltration and reabsorption. This mechanism moves water and dissolved solutes together through the pores, driven by pressure gradients. The balance of hydrostatic and oncotic pressures across the capillary wall, known as Starling forces, determines whether fluid leaves or enters the capillary. At the arterial end of a capillary, the pressure typically pushes fluid out, favoring ultrafiltration. By the time blood reaches the venous end, the pressure favors reabsorption, drawing fluid back in. Not all filtered fluid is reabsorbed. However, about 10% is left in the interstitial space and must be returned to the circulation by another route. The third mechanism is transcytosis, which involves large, lipid-insoluble molecules such as certain proteins and hormones being transported across the endothelial cell inside vesicles. While this is a relatively minor pathway in terms of quantity, it is vital for specific molecules that cannot cross by other means. The lymphatic system, the accessory to microcirculation. This brings us to the lymphatic system, which serves as an essential accessory to microcirculation. Its job is to return the small amount of fluid and any leaked plasma proteins left behind in the tissues back into the blood. Lymphatic capillaries, which are highly permeable, collect this fluid, which then passes through lymphatic vessels and nodes for immune surveillance before eventually emptying into the subclavian veins. The lymphatic system plays a critical role in preventing the accumulation of excess fluid in the tissues, known as edema. If the lymphatic system is compromised, such as by certain infections or after surgical removal of lymph nodes, severe swelling can develop. Summary: to sum up, the microcirculation is an exquisitely organized and dynamically regulated system, finely tuned for efficient exchange. Arterioles control how much blood is delivered to each network. Capillaries serve as the main site of substance exchange through diffusion and bulk flow governed by specific pressures, and the lymphatic system cleans up the leftover fluid. Together, this intricate network ensures that every cell in the body receives oxygen and nutrients while waste products are efficiently removed. This is the remarkable choreography that powers life at the cellular level. Thanks for tuning in. If you enjoy our content, please like, share and hit the subscribe button. It's the Everything Surgery Podcast. Dissect the extraordinary.